everyone. I hope all of you are doing well today. And I'm Iresha. I'm a PhD student at the University of Saskatchewan. So today I'm going to present about our study on antimicrobial immunity involves immuno-metabolic regulation induced by CpG-ODN in broiler chickens. Actually, we all know that bacterial diseases like E. coli, Salmonella, these diseases cause a number of problems in chicken industry. And usage of antibiotics to control these infections has become questionable nowadays. And chicken farmers of Canada have already eliminated preventative usage of antibiotics, belongs to category 1 and category 2. And category 3 also, they are trying to eliminate in near future. Therefore, developing alternative strategies has become essential to protect our chicken industry. So speaking of alternative, this is where our study is connected to the poultry industry. We are working on a molecule called CpG-ODN, which is a synthetic DNA molecule. And naturally, the CpG motifs are present in bacterial DNA. And the CpG-ODN, the synthetic molecule, has ability to stimulate the immune system of chicken without producing any diseases. Therefore, we get that benefit and then trying to use it as an alternative to antibiotics. In the structure of this CpG, you can see here, it has cytosine, guanine, nucleotides, and there's a phosphorothioate backbone. The CpG-ODN within the body of chickens is identified by antigen-presenting cells like heterophils, macrophages, and these cells have TLR21 receptors on their endosomal membrane. So upon this recognition, the cells are stimulated to produce inflammatory cytokines like interferons, tumor necrotic factor. So it initiates the protective response. When we consider major energy producing pathways of immune cells, we can identify cellular glycolysis, Krebs cycle, and electron transport chain. We know that immune system composed of number of different immune cell populations. And even one type of cells have different metabolic status. As example, T cells. It has naive, activated, effector, memory type of cells. So these cells have different metabolic activities. By measuring these major antimicrobial pathways, like energy-producing pathways, mitochondrial respiration and cellular glycolysis, we can have idea what is the energy status of these cells. In our lab, Gomes lab, we have been studying number of studies to prove that CpG-ODN can protect birds against number of bacterial infections. This is one of the example. Here they have proved that CpG can protect broiler chickens against lethal doses of E. coli and Salmonella infections. And this is another example which proved that increased interferons and regulatory cytokine profiles in CpG injected birds, especially in lung and spleen samples. And another study, a recently published study from our lab, have proved that it increased the survivability of birds against E. coli infections, as well as it can alter the serum metabolites level in the body. So up to this point, we know that CpG can protect birds against major bacterial infections. But in this current study, we wanted to go in more detail and then identify how it affects at subcellular level, so we can have a broad idea. So our hypothesis was is that CpG induced the immune system of chickens, thereby it altered the cellular metabolism. So we had 3 objectives. First, to study the major metabolic pathways behind the immune protection mediated by CpG-ODN in broiler chickens. And the second, we wanted to quantify cellular glycolysis and mitochondrial respiration of immune cells following CpG-ODN administration. At the third, we identified which cell type plays major role behind this protection. So at the beginning, we didn't have any methods to quantify this cellular metabolism because it's very rare in chicken industry go to that kind of details and subcellular level, especially real-time measurements. So we use this method, which we can measure the real-time cell metabolism in live cells. So we use this C-host XFP analyzer. We have this machine in our lab for the first time in Saskatchewan. So after optimizing different methods, we used mitochondrial stress assay to measure the electron transport chain of mitochondrial activity, like this is the electron transport chain you can see here. So we use this to measure the mitochondrial stress assay. And the other one, we used glycolytic rate assay to measure the cellular glycolysis. So briefly, we added different inhibitors and stimulants to stress down these pathways. Therefore, we can quantify these energy producing pathways. So this is the laboratory process we optimize, and like briefly, we isolated PBMC layer from blood, and then cells were processed to quantify mitochondrial respiration and cellular glycolysis. And to identify which cell type plays major role from B and T lymphocytes, cells were stimulated with pork-wheat mitogen, which is a mitogenic for both T and B lymphocytes, and PHAP, which induced the cell division and mitosis of T lymphocytes. So after stimulation, these cells in vitro, the mitochondrial stress assay was conducted to quantify the oxygen consumption rate of these cells. So this is the experimental design of our this study. We had 5 broiler chicks per each group, and these were day 1 old broiler chicks. And we had 3 groups. First group received 2 CpG doses, and here 1 CpG dose and saline control as a control group. The CpG was injected using intramuscular routes. Then upon the CpG injections, we have collected samples at 5 different time points. As you can see here, up to 21 days. As a longitudinal study, we used the same set of birds. The samples were peripheral blood. So in the results, so you can see mitochondrial stress assay, mitochondrial respiration results I'm showing here. And these 3 graphs, first graph, basal and maximal respiration, and this one shows the spare respiratory capacity, and this is the ATP production rate. So according to these graph, you can appreciate that CpG received group, here you can see, significantly higher OCA activity in all 3 graphs compared to the saline control group. Actually, we have seen this significant activity at 4 different time points up to 14 days. In the glycolytic rate assay, we have seen a nice separation between 3 groups. Like you can see CpG 2 dose and 1 dose groups. So this nice separation actually we have seen in all the time points, but we saw this significant difference at 48 hours time point only. So this eco activity, we statistically analyze further to get the compensatory glycolysis of these live cells. So here you can see the compensatory glycolysis. So we saw this significant difference at the 48 hours time point in the CpG groups compared to our control group. So in vitro stimulation with pork-wheat mitogen, PHAP, with both 2 groups, you can see here, this is the basal respiration, maximal respiration, and spare respiratory capacity. And according to these results, you can see the pork-wheat mitogen stimulated group had significantly higher OCA activity or mitochondrial respiration compared to PHAP group and no stimulant cells, especially in CpG group. But here you can see in the saline group, we haven't seen any differences. Therefore, this is likely the B cells acting behind this activity. So with that, according to our results, we can conclude that CpG increased the cellular glycolysis of immune cells up to 48 hours. And it increased the mitochondrial respiration of immune cells for prolonged period, up to 14 days. when we compare the lifespan of broiler chickens, it's for a very long period, and it covers first 2 weeks of their life, which they are more susceptible to major bacterial infections. And these cells induced by CpG-ODN likely favor the mitochondrial respiration over cellular glycolysis. And the CpG increased the survivability of B cells for a prolonged period, it indicating phenotype of memory B cells. So these novel methods we optimize and develop in this study will be beneficial for future studies if you want to study like metabolic pathways of live cells in real time. So you can use these methods, and it's very beneficial for our future in research, especially in poultry industry. And by providing this information, we can increase the utility of CpG-ODN to protect our broiler chickens and chicken industry, especially from bacterial infections. So with that, I would like to acknowledge my advisory committee, Dr. Susantha Gomes, my supervisor, and Gomes Research Group, Western College of Veterinary Medicine, Animal Care Unit, GMP, and Clean Path Laboratory. Thank you very much. And our funding sources, University of Saskatchewan Graduate Scholarship, NSERC, Chicken Farmers of Saskatchewan, and Canadian Poultry Research Council, and Western Economic Diversification of Canada. So thank you very much, everyone, for your attention. If you have any questions, I'm happy to answer. Thank you.
Thank you very much, Iresha. There's already questions awaiting you in our Q&A section, so I'll get started with the first one. Well, thank you for your excellent presentation. The person has a question about the number of birds that you've used in your experiment. You used 5 birds per treatment. So do you have any birds which died during your experiment?
Yeah, it's actually we always check the mortality, and at the end we did the postmortems and everything. No birds died, and at the end we check the lesions and everything, whether they get sick or anything. But they all were like a very good condition, so it's no dead.
If I can have a question on my own, you've taken blood samples or serum samples at 24 and 40 days, 48 hours post-hatch in your chicks, which are really small at that time. So what was your, what was the amount of blood collected, and what site did you use for this collection?
Yeah, that's a good question. Like it was hard at the beginning because we needed the enough amount of blood volume. Usually, sorry,
what is the amount that you? That was
yeah, it's around 1.5, 1 to 1.5 we collected, and then usually we actually why we use 5 birds here. We need around 3 ml to process this experiment, but from each bird, like they are very small, we can't take 3 ml. Of course, because we are very small. Then we take the same equal amount of volume from each bird, around 1 ml. Then we pool the blood, and then we process this like a laboratory procedure at the end, like as one sample from 5 pool.
Okay, thank you. So,
and yeah.
So if you pulled them early on, did you pull also the samples afterwards, or since at older age 3 ml at the end was probably enough to, but were all the samples pulled all the time?
Yeah, exactly. We optimized this several, like we have repeated this several times, whether it's affect the, the cells get active, and then sometimes metabolism change. But we did the repeated experiments around 5 times. Then we confirmed that we get every time same results. So the pooling does not affect the results. So we made sure that, yeah.
Okay, so your results are not an average of what you showed on the graph is not an average, but a single result?
I'm showing the pool sample from each group, yeah.
Okay, there's another question. Mr. Lee is asking about the abbreviation PWM.
Yeah, it's refers to pork wheat mitogen, which is a mitogen for both T and B lymphocytes. So we use to stimulate in vitro that component.
Okay, well, thank you very much for your questions and your presentation.